Doctors have known for many years, 50 years, patients that have this specific type of skin cancer because they categorize patients with this type of skin cancer when they look at a biopsy under the microscope and they see a specific type of chromosome damage. Now, normally mutations in DNA are invisible to a microscope because they involve just a single letter change, which is tiny and not visible under a microscope. In the case of this specific type of skin cancer, however, there is a specific change in chromosomes that are visible under the microscope. And the change is that two chromosomes get torn apart. So here's one, and here's the other. Maybe I can color it differently. The mutation, this type of mutation is where the chromosomes are actually torn apart and by accident get glued together to create a translocation. So a specific type of mutation called a translocation, where this part of the chromosome is joined to a different chromosome. And here's the description. This skin cancer is associated with chromosomal translocation where chromosome number 17 is translocated to chromosome number 22. Humans have 23 pairs of chromosomes, and they are named based on their length. The longest one is called chromosome number 1. And the shortest one are the X and the Y chromosome. That. So we have chromosomes 1 through 22 and 23. Then, so there is a specific chromosome, chromosome number 17, that gets damaged at a specific location and translocated to chromosome number 22 that also gets damaged at a specific location. And so doctors could see that under a microscope for years and know that certain patients had this type of skin cancer. That was part of their diagnosis. And then in more recent years, scientists have been able to zoom in not with a microscope but with biochemistry and can now read the DNA letters, which are composed of A, G, C, T, and we'll just start to learn about in our course now. So there's a pattern of A, G, C, T letters, and that includes for coding for proteins like PDGF. And it turns out that the breaks in these chromosomes occur in the PDGF gene for this skin cancer. And the PDGF gene is torn in part and then glued together with another gene. And here are the two genes involved. One is called COL1A1 and one is called PGF-beta. Here's a map of the two genes. So DNA is long strands, and here's a long piece of DNA that is for a gene called COL1, and then here is the other long piece of DNA that codes for PDGF. And scientists have mapped the breakpoints, and what they found is that for the mutation that gives rise to this cancer, there's a breakpoint that occurs in a PDGF gene and then in a variety of locations in the COL1 gene. As a result, we get two genes put together. Both of these genes code for proteins, and this just shows a map for different patients. So these are actual numbers of patients that had mutations that caused the break to occur at various places along the region of the DNA that would code for the protein. This part actually represents the RNA of the genes and shows that part of the RNA comes from the COL1 gene and part comes from PDGF. So we already know something about what PDGF does. So when the protein gets made, it can be released from platelets, bind to the PDGF receptor, and stimulate the cell cycle in skin cells. So this is the functional part of the PDGF gene. Now, what is being joined to it that is going to lead to cancer? What is this gene? And the answer is this is a gene that codes for collagen protein. And collagen protein is talked about in our chapter 3 in our book. It's a protein that's found in connective tissue like cartilage. And there are multiple COL1 genes in our body, in our DNA genome. There are multiple collagen genes. This gene is made in the skin. And so the next thing that we're going to start to learn about is that we have in basically all our cells the same DNA, but specific proteins are made only in specific cells. For example, all our cells have the insulin gene, but the protein, the RNA and protein, but insulin, RNA and protein are only made in the pancreas. So we need to learn about that. And that's a story that appears in chapters 15, 16, 17. So that's why we're going to read and learn about cell communication, the cell cycle. And then we're going to jump ahead to those because we're going to start getting very interested in the control of gene expression, the regulation of that information, and that it's only made in, that insulin's only made in the pancreas. That all the cells in our body have this collagen gene, but this collagen gene is only made in the skin. And it's only made in the skin because in addition to the protein coding region, there's a region up here called the promoter that regulates where the gene gets made. And the problem in these cancer patients is that this part of the collagen gene has gotten torn and glued to this part of the PDGF gene. This part of the PDGF gene codes for the protein. This part of the collagen gene regulates where the protein gets made, and this says make the gene in the skin, and what gets made in these cancer patients is PDGF protein gets made in the skin. Now remember we said normally PDGF gets made in the bone marrow. And when it gets made in the skin, what happens? So in this damaged gene, the promoter activates gene expression and the PDGF gets made. It gets made and it gets made in the rough ER. But in this case, instead of just being held in the vesicles like it is in the megakaryocytes in the bone marrow, in this case, this collagen gene protein normally gets released from the cell. And so when we have this joined together in the mutant cancer patient, the PDGF protein gets released from the cell. And that's a big, big problem because that's where the PDGF receptor is. And so we have now the signaling factor made by the skin cells, and the signaling factor gets released by exocytosis, the way we've learned about. That's the normal way that collagen gets released from the cell, and it turns back and binds to the receptor and activates the receptor, and this is called an autocrine, so a self-stimulating pathway. And it's an example of feedback, and this is an example of positive feedback, where the cell is going to stimulate its own synthesis. It's as if this cell thinks that there are platelets releasing PDGF in the area and that there must be a wound. And this skin cell is growing fast trying to heal a wound that isn't there. And that's what's happening, is we have cells growing out of control and growing out of control because of this event here. Okay? And so we can go and fit this into, this is just a figure from another book showing the idea that the protein is made of both collagen and PDGF. So it's a nice looking protein made up of, you know, the 20 common amino acids, but it has this kind of hybrid characteristic. And we can go to this and we can just plug into, back to chapter 11, this story, and it's the story of PDGF signaling still through the natural PDGF receptor and activating a kinase and activating phosphorylation, and ultimately leading to a cellular response, which is the stimulating the cell cycle. So in the next video, we're going to start talking about the cell cycle, because this is what's going to happen, is that this cell cycle is normally paused at this G1 to S transition, and it is waiting, and this is called start, and the response that the PDGF receptor activates, the phosphorylation, is going to turn this light green and allow the skin cells to quickly go through start and quickly advance through the cell cycle. So we'll talk about that in the next video.